Hello, my name is Candace Ford, and in this video we'll explore the cellular mechanisms of embryogenesis. We'll begin with an analysis of determination, which leads to differentiation. We'll explore both embryonic and adult stem cell characteristics before diving into cell signaling via induction. We'll follow our differentiating cells as they migrate throughout the body, and finish up with a review of apoptosis. In this video, you will learn about the cellular physiology that drives determination and differentiation. The changes we learned about in structural embryogenesis come about as a result of coordinated cell signaling, which follows differential gene expression. By the end of this video, you will learn how to answer questions like the one on the screen that ask about cell signaling and other cell-level developmental processes. Before determination, a cell may develop into any cell type. So, at the very beginning, these cells are unspecialized and thus unable to function to support an entire organism. We can think of the body as a huge factory. There are lots of employees and lots of different jobs to do. The body simplifies this huge undertaking by a division of labor. It sends groups of employees, or cells in this case, to complete specific tasks. Thus, with many groups of employees doing different jobs, our factory is able to function smoothly. These groups of cells, although they have the same genes, end up doing different jobs through selective expression of key genes. Cell determination is the first step in this division of labor. When the zygote first begins to cleave, proteins and mRNA transcripts may be unequally distributed throughout the parent cell. Therefore, when the cell cleaves, the two daughter cells end up with different mRNA and protein compositions. As you can see, the cell on the left already has a transcript for gene A, and the cell on the right has a transcript for gene B. While these early cells have different transcripts and protein compositions, the difference in gene expression is reversible, and these cleavages are indeterminate. However, cascades of transcription factors will be triggered later on, creating irreversible gene expressions due to a changed protein pool. Now our two cells not only have different transcripts, but different proteins. The different protein compositions change cell behavior and signaling, irreversibly changing gene expression. Now the different cells have different levels of expression of each of the genes. At this point, differential gene expression has determined the pathway of a cell. The logical next step, then, is to follow the path. A cell starts to act differently from the basic cell of the zygote, moving towards a specialized job. These cells begin to develop into tissues with different functions. To rehash these points, cell determination is when a cell chooses, based on gene expression, a specific lineage. Cell differentiation, then, is the beginning of this lineage. Cell morphology and behavior change as a cell begins to move towards a specific fate. A way to remember this for test day is that cells know before they show. Determination begins first, followed by differentiation. Determination continues during differentiation as the possibilities for cell fate narrow. Epigenetics is the process through which cell determination is accomplished. If you remember from earlier, cells take on different jobs based on selective expression of genes. Different cells produce different proteins and so can have different functions. Genes are turned on and off by the addition and removal of chemical tags. Methylation, which is the attaching of a methyl group to the cytosines in a strand of DNA, turns off genes. Special proteins recognize these methyl groups and recruit binding proteins that prevent this part of DNA from being transcribed, thus turning the gene off. Methylation is used in the processes listed on the screen. Another way to affect gene expression is acetylation. Histones are the proteins that make up the beads in the beads on a string model of condensed DNA. A protein places an acetyl-CoA on the histone core, decreasing the histone's affinity for the DNA. When the histones can't hold on to the DNA as tightly, it allows polymerases access to the DNA to transcribe the gene, thus turning it on. To simplify this, just remember on test day that access to DNA decides whether a gene will be transcribed or not. Embryonic stem cells are the components of the earliest stages of development. Each embryonic stem cell can differentiate to become any cell type in the body. The range of possible cell lineages is called potency. A totipotent cell can become any cell type. Pluripotent cells are slightly more differentiated, and an example of a pluripotent cell is a mesodermal cell in a gastrula. A mesodermal cell can become any variety of cells in systems like the circulatory system, musculoskeletal system, or anything else the mesodermal germ layer becomes. And becoming more differentiated, we end up with multipotent, then oligopotent, and finally unipotent cells. Unipotent cells can only form one type of cell. So from totipotent to unipotent, we've gone from a cell that can become any cell in the body to a cell that can become only one type of cell. So remember for the MCAT that the less differentiated a cell is, the greater the number of fates it could possibly have. Embryonic stem cells are isolated from the inner cell mass of a blastocyst. This is the point where cells have undergone indeterminate cleavage and can still reverse any differential gene expression. Isolating these embryonic stem cells requires destroying the embryo and is thus very controversial. When we're studying adult stem cells, it's important to remember that adult only refers to cells that are no longer embryonic. These stem cells must be present in the body throughout life to aid in recovery from life's normal wear and tear. So let's explore this idea by revisiting spermatogenesis. Usually, we focus on the meiotic division a spermatogonium undergoes. However, these spermatogonia also undergo mitosis, otherwise we would run out of sperm cells. Once these spermatogonia undergo mitosis, they can then go through meiosis. So, mitosis of these adult stem cells is critical to maintaining a spermatogonium cell population in the testes. We can collect adult stem cells from various tissues, and they are typically multipotent, but scientists are currently exploring the possibility of inducing pluripotency in these adult stem cells. This means that a sample of tissue from an individual could conceivably serve as a source of tissue for that individual, so we wouldn't need a tissue donor. Using an individual's own stem cells to create tissue for them would also greatly reduce the possibility of the body rejecting the tissue. So this is an exciting field of research. Induction is communication between cells that are close to each other. These cells talk to each other via chemicals, and the chemicals can tell a cell to begin differentiating. These chemicals are called morphogens. This induction allows more complex structures to develop, like the optic nerve. Some developmental results of induction are the optic vesicle developing into the lens, the notochord developing into the spinal cord, and limb buds developing into whole arms. Different distributions of morphogens, which were those inducing chemicals, can cause different cells to differentiate, as illustrated in the figure to the right. Let's go more in depth about the different kinds of cell signaling. These chemical signals travel variable distances to affect their target cells, and we should be familiar with the different classifications of cell signaling for the MCAT. Autocrine signals stimulate the same cell where the signal originates. Juxtacrine signaling means that the two cells are connected and the chemical travels through this junction. Paracrine signaling occurs when a chemical travels a short distance to affect a nearby cell, and our little chemical is going to leave the cell on top and travel all the way down to that fourth cell. Endocrine signaling refers to the longest distance a chemical signal can travel. These chemicals travel via the bloodstream, shown in pink on the right, to affect far-off cells. So now we've explored how these embryonic stem cells become different cell types, but how do they get to where they need to be? The cytoskeleton actually pulls cells into the appropriate anatomical position. However, something needs to tell these cells where to go. Chemical or mechanical signals from other cells or the extracellular matrix tell these differentiating cells where to migrate. An example of this migration is neural crest cells. If you recall from structural embryogenesis, neural crest cells differentiate to become the peripheral nervous system, pigment-producing cells, and some glia, so they have to migrate all over the body. Cell migration occurs in grown individuals as well during recovery from injury or in reaction to foreign bodies. At the other end of the life cycle of a cell is apoptosis. Apoptosis, or programmed cell death, occurs throughout development and is an essential part of the normal cellular function of an organism. This is also a really great way for improperly formed cells to remove themselves before they can damage the organism. Apoptosis can be triggered either by internal or external signals, and during apoptosis, DNA condenses into chromatin, and small pieces of the cell break off into what's known as apoptotic blebs. These blebs contain the chromatin and other components of the cell. Other cells would chew up these blebs via phagocytosis, and the contents will be harvested for resources. The main takeaway here for the MCAT is that apoptosis is a positive thing that is helpful to the developing organism. In contrast, the processes listed on the screen are not good things for the organism. Let's try some practice questions to test our knowledge. This first question asks for the classification of cell signaling between two neurons. Take a second and pick an answer. In this situation, we have two cells that aren't in contact, so this immediately eliminates autocrine and juxtacrine signaling as options. This signal is not traveling through the bloodstream, so our answer isn't endocrine either. This type of cell signaling is paracrine, and we have two nearby cells communicating. Let's try another question. Here we are asked about the type of cell potency before and after a process called compacting. Take a second and pick an answer. What process is going on during compacting? Determination. So, our cells are beginning to narrow their options in terms of cell fate. At the start, they could become any type of cell, which is a characteristic known as totipotency. And after compacting, they've begun to limit their options just a bit. Our cells begin as totipotent and then become pluripotent. As a quick note, C is incorrect because multipotent cells are far more restricted in terms of the number of cell types they can become, and we're only losing a bit of potency as a morula. Let's finish strong on our final practice question. This question asks us to associate a developmental mechanism with problems during pregnancy. Take a second and pick an answer. So we're asked to find an answer that relates gene regulation to environmental factors during pregnancy. Answer choice A is out because this has nothing to do with environmental factors, although Barr bodies are certainly a method of gene regulation. Answer B connects the idea of environmental factors and epigenetic markers, which we learned are a method of gene regulation. If you remember turning genes on and off with methylation and acetylation, this is what epigenetic markers are. So B is precisely our answer. C merely describes induction, and D describes differentiation, topics that don't really answer the question posed. We explore the different fates developing cells may have. These cells have to do four main things. First, they must specialize. This specialization is accomplished through differential gene expression and turns our primordial cells into specialized workers. Second, our cells must communicate. Cells talk to each other throughout development and throughout life. Third, as these cells are differentiating and communicating, they must migrate throughout the body to arrive at their appropriate anatomical location. And finally, these cells must, at some point, die. Their deaths help prevent problems from occurring due to damaged or malfunctioning cells. This apoptosis is a normal process for healthy organisms. In this video, we examined embryonic development from a microscopic level, which can sometimes be confusing. You should come back to this video as often as you need to in order to instill this knowledge and fully prepare yourself for test day. Thank you.